Hi, my name is Mary Park. It is week 5, lab 8. Um, it's on biochemical testing for microbial identifications. So, uh, in this experiment, you will be incubating S. cerevisiae and S. epidermidis in fructose, glucose, and mannitol broth, and to categorize each microbe based on the metabolic properties. So I'm going to show you right now, and obviously the experiment has been already done. So what you'll see is yellow tubes, but technically it was red at one point, which I'll show you the results that I have turned in. But I want to show you exactly how the tubes would be labeled. So as you can see, these are the tubes that are labeled. Okay. Um, so you want to label all the tubes, and then you want to make sure you insert the Durham, which is already in there, that into each fennel broth vial, so that the opening of the Durham tube points towards the bottom of the broth vial. And then you want to obviously incubate it for 12 hours. And my incubation spot is in the pantry in a box. Okay. And the part 2 of it is called the carbohydrate fermentation results, which I will be sharing the results with you in the second video. Okay. What I'm going to show you next is antibiotic sensitivity Kirby-Bauer diffusion test, and with that experiment, the Kirby-Bauer diffusion test is with the Staphylococcus epidermidis, and we'll be using 3 antibiotics, which is the penicillin, novobiocin, and the gentamicin. And the purpose of the experiment and the agar plate was made ahead of time. So what I'm going to show you is the end of result, what I did already, but then I'm also going to show you the result that I had turned in. So what I would be doing is, obviously I would pipette, and then I would put it in the agar, and then you'll see how it looks like. I would have to obviously put 3 lines in the agar and labeling it to make for the antibiotics. I would put 8 drops of each of the S. epidermidis on the agar, and then I would make sure that I had spread it evenly, which I'm going to show you right now how it looks like. This is what it's going to look like. And then I would use like a tool, which is an L-shaped spreader, to spread, making sure that I do spread it evenly, the broth evenly across the agar, so that the entire plate is coated with an even layer. And then I'm going to have the agar with the lid side up approximately 1 to 2 hours until the broth is absorbed by the agar. And then once the broth has been absorbed into the agar, I'm going to remove the plastic tweezers from the alcohol, shake it, and dry it. And then using the tweezers to remove the penicillin disc from its plastic bag. And I would place it on, if you see it, right there. And then the other part of it is I'm going to collecting the data from the Kirby-Bauer diffusion test. So that this one's investigating the penicillin research with the fungus penicillin, and observe this fungus with a microscope. After collecting the data from the Kirby-Bauer diffusion test, after 12 hours, as a white film or lawn of layer covering the agar, if no growth is observed, then we want to go ahead and continue the incubation for another 12 hours. So without removing the lid of the agar plate, you want to, I'll be measuring the zone of inhibition around each disc, by placing the ruler over the center of each disc and measuring the diameter of the zone with no bacterial growth. So the next video will show you how my actual results will show. Thank you.